Hey guys, it's David here, and I'm going to be talking about jumping genes and a bit of a background on DNA. DNA stands for deoxyribonucleic acid, and DNA is an instruction molecule that all living things have, let it be you, me, plants, or animals. DNA stores genetic information which is passed on by the parents, and the nucleotides are actually the building blocks for DNA. Now, DNA has three parts to it: a ring-shaped sugar, a phosphate group, and a nitrogenous base. The shape and structure of a DNA. DNA is said to have a double helix shape or a twisted ladder shape because, well, of its shape. The structure is not that hard either. The blue part right here, the sides, is actually made of phosphate group and the sugar, while the middle, or the rungs of the ladder, is actually the nitrogenous bases. Now, if you look carefully, there's only four different kinds of bases: T for thymine, A for adenine, C for cytosine, and G for guanine. Well, so what does this DNA code for? A gene is a long length of DNA that contains the instructions to make a chemical for the body. The DNA in a gene usually codes for a protein. So where is this valuable DNA stored in the cell? All DNA, all chromosomal DNA is stored in a cell nucleus, which is separated from the cytoplasm by the membrane, as you can see in this picture. Alright, so one of the major questions is, what is DNA replication? Just like its name, DNA replication is just when DNA just replicates itself. During S phase, the double helix just unwounds, and helicase, an enzyme, just roots up, just splits the DNA in half. Right there. Simpler picture is right here, and this is the enzyme right here. And each strand acts as a template, and the bases, which is free, like flowing around, just like match up to synthesize the new partner strands. This is Helicase can simply just rip through these because the hydrogen bonds are pretty weak. All right. So as I mentioned before, DNA and genes are related, but how are they related with chromosomes? We'll just look at this picture. Like I said, a long stream of DNA equals a gene, and a long gene actually equals a chromosome. And to finish off the picture, a chromosome, few chromosomes are in cells, and a lot, billions of cells are actually in humans. Alright, now since we covered DNA, genes, and chromosomes, what about we talk about proteins? Protein, what is it? Protein is actually a molecule made from amino acids, like I mentioned before. We can think them as workers. They can be the workforce that actually that gets everything done. Proteins break down everything, such as food, releases energy that's not needed anymore, transports, organizes. Sometimes, well, often, proteins are the useful chemicals we need. So, how does DNA code build a protein? How is protein built? Now, this process is called protein synthesis. This consists of transcription and translation. You can see in this picture, this is transcription, this is translation. This is another picture of transcription. And now the transcription is basically the process where RNA is assembled from a DNA template. The enzyme helicase unzips the DNA, like DNA replication, and free RNA nucleotides just pair up with complementary DNA nucleotides on one of the DNA strands. When the base pairing is completed, the mRNA, messenger RNA molecule breaks away, and then DNA just rejoins again. The mRNA just leaves the nucleus, as you can see in this picture, and goes out to the cytoplasm, which is where the translation takes place. Alright, so the ribosome surrounds the mRNA, which has a genetic code from the DNA, and it's actually being matched to a specific tRNA, which are these, that actually are transferring a specific amino acid to a growing polypeptide train. As you can see right sometimes called a chain. I like to call it a train because it looks like a train. Anyway. Phew. Well, that was a lot about DNA. But what does this have to do with anything about jumping genes? Well, it has a lot to do with jumping genes. Just hold on a sec. Here's a background on jumping genes. In 1940s, Barbara McClintock found that genes could jump around. She won a Nobel Peace Prize for this, by the way. Anyways, just like its name, jumping genes are just genes that have this unusual ability just jumping around and messing up the order. Nowadays, jumping genes are also known as transposons. Transposons are basically sequences of DNA that can move or transpose themselves to a new position within the genome of a single cell, in a single cell, which means sometimes it moves location in the chromosome itself, but sometimes, rarely, the gene can actually pop up from one chromosome and go to a whole different chromosome. Yep, and this can cause major problems, such as mutations. Now, the mechanism of transposition can either be copy and paste or cut and paste. So, studies nowadays show that all living things actually have transposons, which also means that this is not a disorder. Yes, it can cause mutations, but it's still not a disorder. It can either cause a mutation, help the victim, or actually inactivate the gene. You can help the victim because sometimes it's actually medically used to confer a new function or replace a defective gene. But of course, mutations can affect the host. First off, when Miss Barbara was experimenting on jumping genes, she used coins and found out that jumping genes could disrupt genes and lead to spotted kernels. Now, in people, there are diseases and mutations caused by transposons, which include hemophilia A and B, disease that you can't move, muscle disorders, and sometimes predisposings to cancer. Yep, it's pretty intense, and jumping genes are actually pretty important in our lives. Anyway, in conclusion, I hope you really liked my presentation, and thank you for watching.